Welcome to the another class of AP Biology where we will be talking about the biological catalysts today. Have you ever wondered the food that you consume, how you get energy from it? Or whenever the food is being reached in the stomach, how the digestive juices are capable of digesting it and converting it into the different forms of energy. How do you extract energy, how the food is synthesized by the plants. There are so many chemical reactions which carry out where the reactants combine together and form the product, where the breaking and making of the bonds happen. So how these reactions are done. So these reactions are done with the help of these biological catalysts which speed up the chemical reactions and convert the reactants to the products. So let's begin with the enzymes today. So to support all the chemical reaction which take place inside the living system, cells require these specialized proteins called enzymes. And enzymes, chemically, they are made proteins. We know that proteins are made up of amino acids. They lower the activation energy. The reactions which involve the enzymes are generally reversible and they speed up the specific reaction in both the directions since the reactions are reversible. While these enzymes are temperature and pH specific, means that the particular enzyme will have a specific pH and the temperature range. So it will have an optimum activity at its optimum pH, and then this activity will start declining beyond the pH and the temperature range. So they functions best at the temperature which is optimum for it. They form enzyme substrate complex, which is the temporary substance, or you can say that's unstable. And later on, these enzymes are removed or will be recycled or will be taken away, and the reactants will get converted into the product. So they form enzyme substrate complex and they speed up the chemical reactions without getting utilized, which means you can reuse those enzymes for carrying out a new chemical reaction. So it will be free for combining with a new substrate again to get converted into the product. In the process, your enzymes will not get utilized. Now, we know that proteins are polypeptide chains. So you have a polypeptide chain. By the folding of polypeptide chain, some sites will be formed, and this we can consider as the active sites. And active site, what is the importance of active site? This is very interesting to know that these active sites are sockets, and in these sockets a particular substrate will get attached. For example, you have this shape of any shape of the substrate, so you have this kind of active site, then only this will get fixed into this. But if you have any different shape, like you have this kind of shape, so it won't get fixed into this. This is what we call it as the lock and key model. Like my keys cannot open your house, your lock. Similarly, your keys cannot open my locks because the keys and locks are specific. They have particular notch here which will get fixed into this. Similarly, these enzymes have an active site which is specific for the substrate. So this substrate must match to this active site, then only it can get fixed into the enzyme and will get converted into the product. So this is what we call it as the lock and key model of enzymes and substrate. So with the help of this diagram, we will try to understand that enzymes will have an active site which is formed by the folding of your polypeptide chain. So combining the enzymes will get combined with the substrate and will form the enzyme substrate complex. So here your enzyme substrate complex is there by the hydrolysis, the process of hydrolysis. Now, what is hydrolysis? Hydrolysis is a process where by the addition of water, the bonds will be broken down. So here what happens, your enzyme, it gets converted into enzyme product complex, and later on, the products will be released, leaving behind the enzyme. And this enzyme is now again available to react with the new lot of the substrate. So that is why we call it as the enzymes can be recycled. They are not utilized during the process, and thus your enzyme converts the reactants into the product. So your products will be formed. Now, what is the mechanism behind conversion of reactants into products? So this reaction will anyway take place, but the enzyme, what is the function of enzymes? It will speed up the reaction. So when it speeds up the reaction, which means it will accelerate the speed of reaction. We can understand this with a very simple example. Like you go on a trekking, there you get a hill or a mountain. So what will happen? How if you are walking here and you want to reach to the other side of the hill, how will you go? This is a mount in front of you, so you require someone who can, like, either you want this much energy so that you reach to the top, or you require someone who will reduce the height of the hill so that you can reach here quickly. The enzyme will do the same kind of job. It just minimizes the height of the hill for the substrate so that it gets converted into the product very fast. So as you can see here, this is the case where you have without enzyme. The enzyme is not present, just the substrate is there. So substrate has to overcome this much activation energy. Once it crosses this barrier, then only it can get converted into the product. While in the other case, you will find that you have added an enzyme now. So let's look at the progress of reaction. Now what happened? If you look at this part here, the earlier you required this much energy, but now you just require this much energy. How much? This much energy. So now this is your substrate's energy level and this is your product's energy level. Earlier you required this much energy, but now you just require this much energy. So what will happen to the speed of the reaction? It will fasten up. So in presence of enzyme, your substrate gets converted into product quickly because it lowers down the activation energy. So now what is activation energy? So activation energy is the amount of energy which is required for a substrate to get converted into the product. And what enzyme is doing? Enzyme will lower down that activation energy so that your substrate gets converted into the product at a faster speed, as this graph shows that you have, this is the level of substrate, this shows the energy, and this shows the progress of reaction, the x-axis. Now you have a substrate, its energy is here. This red one shows the curve without enzyme, while if you look at this pink one, shows the curve with presence of the in the presence of enzyme or with enzyme. So this is will lower down the activation energy, so your product formation is favored, is facilitated. So now we'll try to understand two things here. Active site. So first we understood the lock and key model. What did it say? Lock and key model says that a active site is specific for the substrate. If it fits into this, then only it will get converted into the product. Now here we have another prospect which says that the active site has an adjustable shape. That means it can make a little bit changes into its shape so that the substrate can fit into the active site. So this, you have this active site where this substrate will get fixed. Now this active site will change its shape little bit to adjust the substrate so that it can convert it into the product. Which means what happened? Your active site has been modified a little bit. So this is what we call it as induced fit model. That means it get induced itself, it makes certain changes so that the substrate can fix into the enzyme. So this is the enzyme substrate complex which has been formed, and as a result you can see the enzyme is unutilized while your substrate gets converted into the product. Now this enzyme can again be utilized to react with the new lot of the substrate. While if you observe here, the arrows you have are in both the direction, reverse as well as forward, which means these reactions are reversible reactions, right? So enzymes can again be converted, react with the substrate and convert it into the product. So there are two models which we understood, lock and key model and the induced fit model. These are the mechanism with which the enzyme reacts with the substrate. Now, what are various functions of enzymes? Enzymes are so important that they give us a lot of chemical reactions happen inside our body. So first is major role of metabolism. Metabolism includes a lot of enzymes. So the metabolism is nothing but the synthesis of food and converting that food into chemical form, right? So the photosynthesis, first thing, with the presence of sunlight, carbon dioxide, water gets converted into glucose, right? So that is photosynthesis. In this reaction, we require a lot of enzymes which convert or fix this sunlight into the chemical energy. Then apart from that, you require signal transduction, digestion. In digestion, we know that there are several enzymes present in the digestive juices, like amylase, you have pepsin, trypsin. So all of these enzyme, lipase, protease, all these enzymes will digest starch, proteins, lipids into the simpler compounds, simpler nutrients from which the energy is extracted out. Signal transduction. Now, what is signal transduction? So, like, how do we communicate with each other? So, we have a power of language. We can talk to each other. We can express our thoughts by writing, by speaking up. But here, the cells, how do they communicate? They don't have a power of speech, power of language. So, they send certain signals. And these signals are nothing but the chemicals only, and these chemicals are nothing but our enzymes. So enzymes facilitate signal transduction. There is an enzyme protein kinase which helps in transferring the signals. Then active transport. So there are certain enzymes which are required in order to transfer of the material across the plasma membrane. You know that plasma, what is the function of plasma membrane? It it forms a barrier through which the materials pass inside the cell and outside the cell. So it controls the entry and exit of the material. So enzymes help in active transport. Then by various biochemical reactions, as I had mentioned, that the photosynthesis, then you have the cellular respiration, all of these reactions are carried out with the help of several enzymes. Enzyme production, the ATP is the energy currency of the cell. So ATP synthesis is being done with the help of the enzymes, photosynthesis, cellular respiration, maintaining the whole body's balance, equilibrium. That is what we call it as homeostasis. That is also being done by the enzymes. So enzymes play multiple roles inside our body, and they help to maintain the orchestra of our body in harmony. This is just to make you understand about the digestive system, where what happens right from the mouth, the food digestion starts with the help of various enzymes in the stomach. Apart from that, the pancreas, the liver, they release certain digestive juices and which contain a lot of enzymes, and they help digesting the food. And the digested food, the nutrients are absorbed in the large intestine. So this is how it helps in the digestion. So just to give you an example, how enzymes are important for us to carry out our various activities. Now, there are certain factors which affect the enzyme efficiency. Now, enzyme efficiency means at in which conditions the enzymes work perfect. So those conditions are temperature, pH, and the substrate concentration. So temperature specifically, I would say you have a specific temperature, which we call it as optimum temperature here, that helps to, here you have this optimum temperature at which the enzyme will work function will function perfectly. Beyond that, what will happen? You can see here their activity declines. Now, what is the reason of this declining? Because you know the enzymes are proteins, and proteins get denatured at high temperature, right? Because they are proteins, so their foldings will open up and their active site won't be there. So no active site will be there to combine with the substrate. So they get denatured and their activity will be lesser. What about pH? So similarly, pH will also play an important role in the enzyme efficiency. So again, you have an optimum pH, beyond optimum pH, like if certain enzyme is there, its optimum pH is 8. Like it functions perfectly near neutral pH. If you go beyond that, what will happen? Its activity will decline. And substrate concentration. Can you guess what will happen if you increase the substrate concentration? You have more substrate, so more product must be formed, right? Same thing happens here. If you have substrates, you keep on increasing the enzyme concentration, the product formation will also increase, but there is a limit because your enzyme is limited. So now what will happen? There will be a stage comes where it becomes steady. Why? Because the saturation reaches here. The saturation reaches. So beyond a certain limit, beyond an optimum condition, concentration, it becomes stable, right? So these are the various factors which will affect the enzyme efficiency. Now there is one more important thing which we consider as the enzyme inhibitors. There are certain cofactors as well. There are certain there are certain coenzymes or cofactors which are also required for to increase the efficiency of enzymes. On the contrary, these are the enzyme inhibitors, which means they will decrease the enzyme activity. They will negatively affect the enzymes. Now, there are two major types of inhibitions. One is competitive inhibitor and another one is non-competitive inhibitor. Now, competitive inhibitor is, as its name indicates, it mimics the substrate and it will compete with this active site. So you have the active site. We know that active site specifically binds with the substrate. Now your inhibitor will have the same shape as your substrate, and it gets fixed to the active site before your substrate comes. So now what will happen? Your substrate won't be able to combine with the active side because it has already bound to an inhibitor. So there is no free space, no active site freely available to bind with this. So enzyme activity, the substrate won't be get converted into the product. While the non-competitive inhibition says that here what happens, it doesn't compete with the active site, but it has another side which we call it as non-competitive inhibitor binding, or you call it as allosteric site. We call it as allosteric site. At this place, it will bind and the shape of active site changes. Because of that, the certain chemical changes will happen. This allosteric site, the allosteric compound bounds. Because of that, the active site changes its shape and there is no more binding. So substrate won't be able to get converted into product. So in competitive as well as non-competitive, in both the cases, the inhibition will happen. Substrate won't be binding with the enzymes active site. Thus, no product formation will happen, right? So this is how we understood about the enzymes. Now let's try to look at the final review, that here what happens, the enzymes are so important that they are present in the cells, plant cells, animal cells, which help in the photosynthesis process, cellular respiration process, so that the energy can be reduced, right? So the enzymes are so important to carry out various activities, to carry out various chemical reactions inside our body. Thank you. Stay tuned.